Good morning, everyone, and welcome to our DeBakey Heart and Vascular Center Grand Rounds. This Grand Rounds is broadcast live via live stream, so we'd like you to interact with us and send us some questions through that and make it live. And today is a special Grand Rounds because it is our William Winters lectureship. It's amazing that it is our 30th William Winters lecture to honor Dr. Winters, who was with us for so many years, no longer with us for the past 3 years. But this is in his honor and his memory because he was really a pioneer in cardiology for the Houston community, among the first to do cardiac catheterization in Houston. He brought non-invasive imaging, echocardiography back in the early 70s in the basement here at Houston Methodist, and also brought PCI procedures at that time, PTCA called. Dr. Grunzig, introduced that. And he founded the Houston Cardiology Society, was president of the college, the American College of Cardiology. And most importantly, an amazing clinician with great bedside manner, a clinical investigator, and a mentor to many, including myself. So, and the lectureship has brought amazing investigators and individuals in the cardiology field. Last year, we had the honor to host Dr. Brownwald. It was a virtual meeting, but it was so nice this year to get back in person and being here. And before we go through the introduction today, I wanted to acknowledge one of our guests today who won't be any guest anymore, Dr. Ardehali, who just accepted the position of a chief of cardiology at Baylor College of Medicine. So welcome to Houston. He gave Grand Rounds a couple of years ago, so it'll be wonderful to have a chapter of collaboration among our institutions, Baylor College of Medicine and Houston Methodist. Today's lecturer is a pioneer in cardiovascular medicine and basic sciences. Dr. Christine Seidman does not need an introduction. Anybody in this room and watching us throughout the world recognizes her name and her husband's name, Dr. John Seidman. They've been collaborators for close to 50 years, right, Christine? And he's her academic partner and partner in life for many, many years, and amazing to see this power couple introduce so many concepts, solve so many mysteries in cardiovascular medicine that not only we are in awe, but I think it's an amazing gift to our patients, particularly in cardiomyopathies. So Dr. Seidman is the Thomas W. Smith Professor of Medicine and Genetics at Harvard Medical School, and investigators for Howard Hughes Medical Institute, and also the Director of Cardiovascular Genetic Services at the Brigham and Women's Hospital. She graduated undergrad from Harvard, went to medical school at George Washington University, internal medicine at Johns Hopkins, and cardiology fellowship at the Mass General Hospital. She and Dr. Seidman are amazing investigators. I think their major contributions are the discovery of the genetic basis of hypertrophic cardiomyopathy and dilated cardiomyopathy, as well congenital heart disease. They built experimental models to understand the mechanism and the pathogenesis of these cardiomyopathies. And if you think, I know we toss around the word translational medicine, just imagine the power of what they were able to contribute. Going to the basic sciences, develop experimental models, animal models of hypertrophic cardiomyopathy and cardiomyopathies, understand the genetic basis, the molecular basis of this. And as we've seen as of late, is develop this into targets for therapy. And this is translational medicine. Nowadays, actually, we have the first drug, thanks to their investigation, that targets the therapy for hypertrophic cardiomyopathy. In the past, obviously, we used available drugs that are not specific for the disease. And I think we owe them a debt of gratitude for all the work that they've done, really pioneering work. Dr. Seidman is the recipient of so many awards, and I will name a few. The American Heart Association Basic Science Prize, given last year, the Joseph Vita Award, the American Society of Clinical Investigation Award, the Passero Foundation Award in Cardiovascular Research, the Bristol-Myers Squibb Award for Distinguished Achievement in Cardiovascular Research, the European Society of Cardiology Gold Medal, and as of late, actually, will be given next month in March, the Valentine Fuster, the new award, Valentine Fuster Award for Basic Sciences, Impact in Cardiovascular Medicine. She's a member of the National Academy of Medicine and the National Academy of Sciences. What a pleasure to have you, Dr. Seidman, with us today. We're honored to have you, and we look forward to an amazing lecture giving us some more insights into the genetic basis and the impact of genetics on cardiovascular medicine.
This is my good friend. Could you tell? I wish my children could think so highly of me as do my good friends. It's such an incredible honor to be here today. I've visited Houston a couple of times over the past few decades, and I must tell you that every time I come, it grows and gets better and better. And for those of you new to the Houston community, this is truly the Mecca of medicine. It is a powerhouse of hospitals, and much more importantly, it's a powerhouse because of all the people here who make it. So with your incredible clinical care, and most relevant to what I'm going to tell you about as well, your ability to do research to change the future of medicine. And that's where I'd like to pose, where I'd like us to begin today. These are my disclosures. None of them are particularly relevant to what I'm going to tell you, but I want for CME informations to have that disclosed. Okay, so for many years now, I've been fascinated with the cardiomyopathies, largely because it seemed to me as a young medical student that the heart was a very simple organ. When it got sick, it did one of two things, like a light switch. It would dilate or it would hypertrophy. That was about it. Figure it out and you'll cure disease. Well, that wasn't quite, as you all know, exactly the case. And so I spent a lot of time really thinking about what I could drive my research focus in, and I became very invested in hypertrophic cardiomyopathy, largely because of the diverse clinical manifestations of this disease, be that really substantial outflow tract obstruction or very subtle forms of hypertrophy that seem to be very well tolerated. And over the years, we collected families, and I went and made house calls and visited people around this country and indeed around the world who had familial incidents of hypertrophic cardiomyopathy. And that led to a really clear revelation that this was an autosomal dominant trait, but with disease-related, age-related penetrance. And so the clinical manifestations were not always present until individuals largely reached puberty. But that kind of information that we were involved in was complemented with a wealth of really phenomenal imaging studies as well as hemodynamics that led to us really to define the recognition of the pathophysiology of disease. With that in place, then, we could set the stage for defining the genetic basis of the disorder. And initially, this required restriction fragment length polymorphism analyses across the genome, and nowadays, obviously, direct sequencing of an exome, a genome, or targeted panels. And we know that there are two major genetic causes, and those include dominant mutations in the myosin heavy chain. Those are all missense mutations, and so it's a single amino acid substitution for the other. And what that does, I'll show you in a minute, is to alter the function of the sarcomere. The other major cause is myosin-binding protein C mutations, and these are not missense. They are a loss of function, and they foreshorten the molecule, and thereby, because it is unstable, reduce the quantity of C protein within the sarcomere. It's about 50% of normal levels. Furthermore, with those genetic understandings, we could begin to build models. And so we made a number of different mouse lines, which expend incredible amounts of money, much more than medical school tuition. And those have allowed us to really begin to understand some of the clinical manifestations in a longitudinal fashion, at least from the point of view of a small rodent. More recently, and with the advent of newer sequencing approaches, we can do two things. The first is that we can validate and further our understandings by directly sequencing human heart tissues that are excised during myectomies or, unfortunately, still in this day, to transplantation from hypertrophic cardiomyopathy, and begin to understand what are the consequences of these mutations on the global sense of myocardial biology. And as all of you know, we've been able to develop a new research model, a cell model, an iPS-derived cardiomyocyte. We don't use, for the most part, patient-derived cells. Rather, we introduce human mutations into particular lines that we've engineered to contain a green fluorescent protein on the N-terminus of titin so that we can image the sarcomere, measure its contractility and relaxation. And it's an awful pretty picture, too, I think. Okay, so with that information, my dear colleague Carolyn Ho has, with collaborations from literally around the world, collaborated to begin to understand what is the natural history of this disease today. I know Gene Brunwald was here not too long ago, and I know he told you the historical context of this disease. But when he saw the disease while at the NIH, they had very little understanding of the adverse outcomes or the opportunity to prevent them, in particular sudden cardiac death. We all were recently aware of the importance of that devastating consequence from the football games over the holidays. And as you know, we now can prevent them if we know in whom to place an AICD or perhaps still use reparative antiarrhythmic medications. So Carolyn felt that it was time to really relook at the natural history. And what you can see from this slide is that you don't see sudden death. In fact, that really remains significantly reduced, which may reflect the advent and long-term use of medications, as well as certainly the placement of ICDs. But what I want to draw your attention to is the panels that show that sarcomere mutations appear to have a greater risk of the really continued problems with HCM, meaning atrial fibrillation and heart failure, as compared to those individuals who have clinical manifestations but do not yet have a genetic basis. Well, this is a dated slide, and as many of you now know, there are SNPs, single nucleotide variants, in non-coding sequences around the genome that are able, when in large accumulation, to have a phenotype that looks very comparable to a dominant mutation in myosin and C protein. The polygenic risk score has arrived to HCM. I'm not going to talk about that right now because we don't have a way to tease out the contribution of those individual loci around the genome, but rather focus on the two major dominant levels. I also want to point out that that graph in the middle shows a blue line, and these are individuals who have clinical disease again and have a variant in a sarcomere protein, but we have insufficient knowledge to understand if it's pathogenic or a relatively personal, private variant that arose within their family, or perhaps within an ancestral population. And so we call these variants of unknown significance, the dreaded VUS in genetic terms. And they remarkably seem to have outcomes that sit between those who don't carry a dominant mutation and those who do. That's something we'll come back to in a minute. With these genetic insights, therefore, we've been able to begin to tease out the natural history in patients, not in animal models. And what we've learned over the years is before there's hypertrophy, which really was the focus of disease manifestations and clinical diagnosis for many years, before the onset of that, we can find hyperdynamic contraction in the left ventricle and as well poor relaxation. My colleague Hugh Watkins has also shown that in those individuals without hypertrophy who carry a mutation, that their energy consumption in the heart is also already increased. Well, that to me was really a revelation because it says that these are the proximal manifestations of these gene mutations. Before the remodeling, you get hyperdynamic contraction, poor relaxation, increased energetic usage. So how can we then take that back to the bench and understand why does it occur? And to do that, we've built, again, these models in mice. And initially, we would isolate single molecules from the hearts of little mice who carried hypertrophic mutations. And using devices that were developed by Jim Spudage and colleagues, we could measure their ability to slide along balls that are tethering a laser trap and be able to measure the dynamic movement across a sliding actin filament. And what we found, quite surprising, is that a missense in myosin increased the rate of velocity of sliding, increased ATP hydrolysis, and increased force production. Fast forward, and those same kinds of experiments were repeated with a little bit different mechanism and different approach, rather, and we found that loss of function of C protein does exactly the same thing. It augments the sarcomere power by increasing the velocity and ATP hydrolysis of myosins. Now, those are in mice and in a very large ex vivo model, and indeed involved only a single head of myosin along those actin filament. We wanted to make sure that was valid, and now with iPS cells, we can, again, with our GFP-tagged sarcomeres, begin to measure contractility. And I'm showing you here a wild type in blue, that's a healthy, normal myosin, a myosin with a missense mutation, and a C protein loss of function mutation. And I hope that you can appreciate, are these beating? I can't see if they're beating. Okay, that the hypertrophic mutations increase dynamic contractility within the heart, and as I'll show you in a little bit, they reduce relaxation. So that told us, without remodeling, we have a fundamental biophysical consequence that increases the power of the sarcomere. Well and good. How does that explain clinical manifestations? Rarely do we think that a heart will be sick because it functions in too well a way. Rather, we think of it being depressed in its biophysical function as causing heart disease. And so what we wanted to do is really begin to understand the other early and proximal manifestations of diastolic dysfunction, because those are the ones that really drive the major outcomes, in particular, the development of atrial fibrillation. If you have poor relaxation of the heart, as this audience well knows, you're going to have an increase in left ventricular and diastolic pressure reflected into the left atrium, passive stretch of that small chamber, and over time, development of AFib. AFib occurs in about 20 to 25% of all patients with hypertrophic cardiomyopathy across their lifespan, which is far greater risk of an arrhythmia than any other condition that I'm at least aware of. So how do we understand diastole? That really is a phase of the cardiac cycle that I really believe has had far too little attention. And there's not a whole lot understood about how it actually occurs at a biochemical and biophysical level. We know for many years of work from the muscle field that when contraction occurs, the contraction reflects the work of two heads of myosin. They always work in pairs. They always hydrolyze ATP as they reach out to the actin thin filament, and contraction is highly effective with these paired myosins. But during relaxation, as it turns out, there are two different phases that can occur, and these reflect conformational changes with each of the two paired heads of myosin that are asymmetric. As you can see in the middle of this diagram, in one instance, the head that has previously reached out to the thin filament has doubled back down to the thick filament, and that X indicates that the ATP hydrolysis pocket is inhibited because of steric interactions. By contrast, you can have another phase of relaxation where both heads have doubled back down and there's no ATP hydrolysis. Biochemists were the first to discover this because they could measure the rates of ATP hydrolysis, and they found them to be, not surprisingly, highest during contraction, but a consistent two-phase step down with relaxation. Well, is that really happening in reality? I had the opportunity several years ago to collaborate with a brilliant scientist from Venezuela, Raul Pedron, who had the opportunity to study a system that was very close to his home as well as to his heart, the tarantula. And he was able to get low resolution cryo-EM of the tarantula myosin locus, myosin molecule rather, in its different phases of relaxation. Now, the tarantula myosin, you would think, is not much related to the heart myosins, but indeed they are approximately 80 to 60% identical at the amino acid level. So we converted his structure into a human structure, and as you can see here, we have two heads of myosin, one green that is reaching out to the thin filament, one yellow that is doubled back down, and when you put it in the context, let's see if I can play this again. And if you, not going to do it. There we go. If we put it in the context of the backbone of the thick filament, it fills the space perfectly. So while it's not a perfect representation of myosins, it's pretty darn close. With this limited information, we could then understand the residues that have to change their position and interact with other molecules in the sarcomere to make those semi-relaxed and fully relaxed conformations. And then we asked a simple question: where do myosin heavy chain mutations fall? And as it turns out, they are highly clustered in the very residues that interact with the other myosin molecules or myosin binding protein C or the like, the interacting residues involved in super relaxation, where both heads are doubled back down. Moreover, when we looked at some of the other structures within this domain, we found that the essential and regulatory light chains are also there, and all mutations that cause HCM in those two rare causes of disease are also within those interacting residues. We don't see any enrichment in the general population of normal variants in myosin, suggesting, in fact, that these have a profound biophysical effect. And perhaps most interestingly, 70% of the hypertrophic mutations change the charge. Now, all of you remember from your basic biochemistry that charges are one way to increase the stability of protein-protein interactions. And if you alter the charge, you can actually repel those interactions. And that's what we think is going on when we have a missense mutation that substitutes one amino acid for another, because they usually come with a charge change. So what that would predict is that our ability for assuming those fully relaxed conformations is inhibited when you have a myosin missense mutation. And that's going to shift the relaxation phase to this intermediate stage in which one is going to continue to reach out to the thin filament and hydrolyze ATP and continue to try and semi-contract. Poor relaxation and increased energy consumption. Okay, so we wanted to formally test this, and to do so, we had to actually be able to measure ATP hydrolysis. And we did that using a fluorescent dye tagged to ATP, and then we would chase our tissue or cells with cold ATP, which is not fluorescently labeled, and look at the rate of decay of fluorescence. And you can see here that the rate of decay is not linear. It comes in two phases: fast hydrolysis, which is when you have that semi-relaxed phase, and full slow hydrolysis when you have doubled down both myosin heads and you can't even bind ATP. And then we asked, is there a physiologic system in which we could measure this that might give us some insights that this is biologically relevant? And again, we turn to a rather interesting animal, the 13-lined ground squirrel. Some of you may read the books of the year in review, and one of the ones that I recently was fascinated to learn about, that the New York Times selected as one of their notable books, is a book about what animals can teach us. And I draw your attention to the chapter on the 13-line ground squirrel. This is a mammalian hibernating animal. So deep in the winters of Boston, not Houston, it's been known for a long time that these animals really shut down their whole metabolic system, and they nestle quietly under leaves or something and drop their heart rates, their basal respiratory metabolic and rates, and as you can see, they assume this lovely little phase of being held in the hand. A colleague, John Stapleton in Canada, has studied these animals for many years and is able to isolate from the hibernating animal tissues that we can then study. They also obviously wake up in the springtime, and they intermittently wake up during the hibernation cycle, something that they call interbout hypothermia, so euthermia rather, where they also assume quite a bit of activity. So with hearts from active animals and those in this interbout period, as well as hibernating, we measured the composition of SRX, DRX, and ATPase. And what you can see here is there is, when the animal is awake during the summertime, more of the semi-relaxed myosins, and when you are hibernating, you have far more of the fully relaxed hibernation, myosins rather. In fact, there's a 40% shift in the proportion of myosin conformations. We think that is absolutely critical for maintaining the low level of energy that is required, and they can mount during hibernation. It makes a lot of sense. I suspect when we're deeply asleep in the middle of the night and our heart rates plummet 40, 50 beats per minute, this is exactly what we're doing. But I haven't yet figured out how to get a little snip of that tissue without waking someone up. Okay, so that's a hibernating ground squirrel. What about people? Well, we weren't initially able to do it in people, but have done so subsequently. But we have been able to use our iPS cells. And when we engineer human mutations into the same background in myosin heavy chain or in C protein and measure their SRX and DRX ratios, we see again that these human mutations shift the ratio so that these cells are chronically always in the semi-relaxed state and hydrolyzing more ATP. There's about a 20% shift in the ratios depending on which mutation you have. Okay, does it do anything? Well, it sure does. So we've taken those same cells and we've profiled their metabolic capacities, either in a wild type or again the same genetic background but with the human mutation. We've measured their sarcomere content and we've measured the sizes of those cells, and all of them show substantial differences. The size of the cell increases, the sarcomere content increases, and they shift from normal fatty acid hydrolysis to glucose utilization. Again, signature of a stressed cardiac myocyte. So with that point understood then, we thought it would be good to return and see, can we have an evidence that this is meaningful to the hypertrophic patient? Not to an animal, but to a real human being. And I returned to those individuals who had those variants of unknown significance. We know that the majority of those are actually in myosins. And the reason for that is a very simple genetic one, that if you have a loss of function mutation, a stop mutation or a frame shift, we know that's going to kill the protein. We have no trouble making that genetic diagnosis. If you have one amino acid substituted for another, it's a little hard to prove that's going to really do something damaging. So what we did is we took all of those variants of unknown significance, and with our knowledge about the residues that are critical for interaction and forming relaxed sarcomere structures, we asked, are those variants of unknown significance in the same interacting residues, or are they elsewhere? And so our hypothesis was that some of those are benign and they don't cause disease, and some of them are really pathogenic and in those interacting residues. And when we did that, we found, yes, some of them were, and then we asked, what are the clinical outcomes of those patients who have a VUS, but it's in an interacting residue? What's their rates of atrial fibrillation and heart failure? And they almost identically mirror what we see in those individuals with a pathogenic mutation in myosin that's been firmly established from familial segregation. So what that says to me is that these VUSs can be informed from our understanding of structural biology and the biochemical consequences of that. and that we now can really risk stratify these patients in a very different way and improve our understanding of those who unfortunately are at highest risk of these adverse outcomes. Okay, so if you can do that, you want to treat it, you want to fix it. And so several years ago, Jim Spudage and Leslie Leinwan, John and I formed a small company so that we could do a high throughput screen. And first you needed an assay, but I've told you over and over and over again that we can measure ATP hydrolysis. And so we decided that would be an effective way to begin the screen. And we went through thousands of molecules and looked only for a molecule that could perturb ATP hydrolysis of myosin molecules. And we found one called MY6461. And as you can see from its structure over here, and more importantly in the schematic at the bottom, when we applied this molecule to myofibrils that we had isolated from animal models, we could see that as we increased the dose of 461, we decreased the ATP hydrolysis. Moreover, when we took that same compound and then applied it to our iPS cells, we found the same thing, except for one big difference. We also found that it restored the normal balance of SRX and DRX. So it reduced the hyper-ATP hydrolysis by shifting back to a super relaxed phase of contraction without having a substantial impact on contractility. Moreover, the beneficial effects of that was that it normalized oxygen consumption and it reduced the hypertrophy in those cells. Okay, what happens in vivo? Well, we had made these mice and we thought it would be very provocative to see if we could treat our animals. And we did that in 3 different myosin mutations that are shown schematically, 403, 453, and 719. 2 of those mutations, 403 and 719, are the ones that most profoundly affected these balance of relaxation conformations. And what you can see on the right panel is that if you have an untreated animal, that these develop left ventricular wall thickness, hypertrophy, by the time that they are approximately 25 weeks of age. When we gave these animals 461 in their drinking water, we found that we could prevent the hypertrophy from ever emerging, and that these animals appeared to be no different from the wild-type litter mates. Moreover, when we then sacrificed them and looked at the accumulation of fibrosis within their hearts, and RNA sequencing confirmed this, as you can imagine, at a molecular level, that the amount of interstitial and focal fibrosis that we see in a hypertrophic heart that is untreated is largely resolved or doesn't even appear. We've not yet been able to show reversal, which is an important step. But if we give this drug early on, we can basically prevent the disease from emerging. Well, not work that I have been doing, but Myocardia went forward with this and developed a drug that was called mavacamten and now has a trade name of Camzyos, the worst name on the planet as best I could think of. And they began to explore how this might benefit patients. And it's an interesting kind of study to think about, and I think one that we all are going to be facing as we look at these chronic, insidious, but progressive diseases. How can you get a fast enough readout to know that your molecule is doing something? You can't wait 30 weeks in a mouse or 30 years in a human to see what happened. And so here was a real opportunity because, as you all know, many patients with hypertrophic cardiomyopathy have outflow tract obstruction. And we've recognized for many years that it's dynamic. It's not the same, millimeters of mercury each time you measure. It fluctuates, suggesting that there's some lability there, which probably has a lot to do with hemodynamics. And so through a series of phase 1, phase 2, and ultimately a phase 3 double-blind study, this was taken forward. And in the ultimate experiment, patients were randomized and blinded by both the patients and the doctors delivering it to ask whether we could alter the outflow tract obstruction in hypertrophic patients. And I show you here the metrics that are being assessed. Yes, they're looking for a reduction in left ventricular outflow tract obstruction, but as well, they were looking for increase in the ability to mount greater maximum VO2 when they are exercising, as well as improvement in New York Heart Association class, and of course, patient symptoms. To me, the most remarkable finding in this study was as follows. One had a very substantial outflow tract gradient. The drug did what we thought it might be able to do, but it did not change ejection fraction, not to any substantial degree. There's about a 4% decrement in comparison to untreated patients. If you can do that, how would that occur hemodynamically? You're not increasing contractility. You're basically improving relaxation. And so the flow out is unchanged, but the gradient disappears. Okay. To prove that, we've looked at a lot of biomarkers as well. And one of the remarkable ones that all of us follow with cardiomyopathy patients is N-terminal BNP. And there was a remarkable drop in N-terminal BNP that stays down through the life of treatment. Moreover, and quite surprising to me at least, was the high sensitivity markers of myocardial stress, troponin I, were similarly remarkably reduced. So all of this predicts that we have the opportunity for the first time to possibly limit the development of atrial fibrillation, and maybe, if those stress markers are telling us something, the progression to heart failure. Okay, so I've tried to tell you, one last thing, sorry. Most recently, people have begun to see, do you actually change morphology in the heart? And the answer is yes. In a very small substudy of MRI of patients who had been on the drug for long term, one found that their wall thicknesses decreased, they became normalized, and their left atrial dimensions also were reduced. Again, supporting that you may in fact be having some reverse remodeling. I doubt, quite frankly, it will go back to normal if you have established fibrosis. As best we know, this is not going to have the opportunity to cure that. That's another problem. All right. So what I've tried to tell you is that these drugs have taught us a whole lot about the biophysics, but that the basic science has driven how we can really think about using that for the benefit of patients. We know now that there is an important component of relaxation that, to my perusal, has not been fully appreciated by cardiologists and researchers. These two phases of relaxation, if we can manipulate them, think what we could do for diseases like HFpEF, if we could increase the ability of that heart to be more fully relaxed. We know that when you don't have that capacity, your hypercontractility and increased use of ATP over time probably takes its toll on the myocyte. And as we know, you can shift to glycolysis for a short period of time, but long term that's not going to propel sufficient ATP generation, and the heart will fail. We have lots we don't understand in the context of even dominant mutations, the troponin mutations, which are less common but very, very impactful, in particular troponin I. Do they have these same consequences? One thing I didn't tell you is that the clinical trial with mavacamten did not genotype patients. And so I think this remains an open question about whether there is some link between thin filament mutations and how the drug works in those individuals. We'd also like to understand if those will, in fact, when provided long-term, prevent the development of AFib and heart failure. And most importantly, can it reverse established disease in individuals? Now, while all of those things are opportunities that are going on in big pharma and beyond my little research laboratory, I think that one of the things we've turned our heads back to is thinking again about the basic mechanism. These are dominant gene mutations. They come in two flavors. They are loss of function for C protein, and they are missense mutation in myosin. The ultimate opportunity of knowing that should be a chance to actually prevent disease for good, to cure hypertrophic cardiomyopathy. And to do that, we've thought a lot about how we might strategize to develop the opportunities to do that. If you have too little protein, you might give back more of the protein from an exogenous delivery method. And indeed, there are companies, in particular Rocket Therapeutics and Tenaya, are trying to give back C protein to individuals who are homozygous for mutations to try and restore that capacity. It may work. I don't know that it will, but I will caution you that C protein is one of those molecules that undergoes phosphorylation with profound impact on its function. So if we have too much, will that be appropriately regulated? It's an open question. Myosin mutations, on the other hand, we know exactly what they do in more detail, I would suggest, than C protein. And while it certainly undergoes post-translational modification as well, those missense mutations we are pretty confident are driving a very specific consequence. So can we correct those gene mutations? Alternatively, could we just silence the mutant gene? We can look into the sequencing databases and ask, are there individuals in the world who have only one gene copy of myosin because of a rare mutation that silences the other? And there are. There are not many of them, but they are participating in big data banks such as NOMAD and the like, which exclude individuals with disease that is really overt, such as hypertrophic cardiomyopathy. So we think that haploinsufficiency of myosin is probably okay. We're testing that in the lab right now. But there's another way that we can think about actually correcting a mutation, and that's come from the evolution of enzymes that have been recognized to exist in bacterial systems for many years. These are called base editors, and they come in two flavors. Cytosine base editors are the ones that naturally occurred, and David Liu and his colleagues at MIT have evolved that to create something called an adenine base editor. What these molecules do is they convert one nucleotide into another without cutting the DNA and substituting in a different nucleotide. They actually are chemically changing it. That's really powerful information because we know if we look across all human mutations that have been characterized by many different physicians and investigators for hundreds of years, that about 30% of them are transitions. And those are the ones that are adenine or cytosine altered to a different base. Okay, so we thought it would be provocative to begin to ask, can we use these in our model systems for myosin missense mutations? And as you might imagine, we picked one of the most well-studied mutations, the arginine 403, because, first and foremost, it is a substitution that is a transition. It has an amino acid change that we know and is adverse and has profound functional changes on SRX and DRX, and we had mouse models with that very mutation in its germline. And what we did initially is to use our iPS cells and to deliver these base editors, an adenine base editor, and to measure contractility. I've told you that's a quick readout. Hypercontractility can be readily measured with our green fluorescent protein. And here I show you a culture, an admix of cells. Some of them are edited, some of them are probably not. But as a population of cells, we see that the hypercontractility in the untreated cells is restored towards normal, suggesting that it's doing something very potent. Well, that's a first step, but we wanted to take it a step further. Let's give it to a mouse. What I didn't tell you is that, unfortunately, these editors are quite large, and they have to be delivered in not one, but two vectors. In particular, adeno-associated vector, serotype 9, is one that we know gets into the heart. Roger Hajar was the first to have shown that in his CIRCA trial many years ago in the human trial, CUPID trial. So we first got that adeno-associated virus and said, can we get it into the heart? We took a green fluorescent protein, our favorite color in the lab, and as you can see, when we injected it intrathoracically, not into a vessel, we got brilliant green colored hearts with very little green in the liver, suggesting there really is organ specificity. But now we need to get two into the same cell to have an editor instead of a green fluorescent protein there. So we made another protein, a red fluorescent protein, and then asked, could we get both green and red in the same heart? And I hope I can convince you from this visualization that the merged picture on the right has many cardiomyocytes with both fluorescent markers. When we look at immunohistochemistry, it really is in the same cell, and it really is in cardiomyocyte because we've driven the expression of these proteins under the troponin T promoter, which is only selectively expressed in cardiomyocytes. Okay, we've now then begun to inject our dual vectors at a single dose, at a single time, early in the life of a mouse, and ask whether we can edit that arginine 403 residue. And when we then sacrifice the animal, take out the heart and sequence, we can see that approximately 80% of the cardiomyocytes, because this is single cell sequencing now, 80% of them, in fact, carry the normal residue only. We've gone on to look to see have we cut or silenced or killed the mutant allele. We have not. And so what that means is that we've really had appropriate base editing. Moreover, when we look longitudinally over time, we can once again prevent the development of hypertrophy so that these animals are comparable to what we see in a wild type in comparison to the untreated animals. And perhaps equally impressively, if you deliver these base editors early on, you can totally silence the fibrosis. What I won't show you is all of the background information too. We don't have liver toxicity. We've looked at serum enzymes. We've looked at BNT. We've looked at troponin T. These mice are perfectly healthy. What this suggests to me is that we are beginning a new opportunity in genetic medicines and cardiovascular diseases. Think how many disorders we know are driven by a single gene mutation. Marfan syndrome, hypercholesterolemia, familial hypercholesterolemia, all of the dilated cardiomyopathies, not all of them, but a lot of them. And if we had the opportunity to really design these powerful nucleic acid therapies to treat either a group, which can be done with when those mutations are clustered in a small segment of DNA, such as RBM20 mutations, or individually, if they recur over and over again, such as the phospholamban mutations that cause dilated cardiomyopathy, maybe we can make a real impact on the lives of our patients and actually prevent disease development. So I would like to propose to you that since we know so much about these genetic bases, it's time for this community to embrace nucleic acid as a new therapy going forward. We have good understanding of the longitudinal histories of these diseases, so we know when to deliver these therapies, at what time in the course of a person's disease. We also know that some mutations are pretty benign and well tolerated for life. They don't need this kind of advanced treatment. But those individuals who really are going to progress rapidly to heart failure or have high rates of sudden cardiac death are the ones that maybe we should be addressing. Moreover, I'd suggest to you that those individuals who are poised to undergo cardiac transplantation provide us an opportunity akin, in my estimation, to a safety trial in a cancer patient with end-stage disease. We could have the opportunity to begin to move these into that group of patients and ask whether, in fact, they are at least safe for the heart that is going to be explanted anyway. There's also rapid emergence of these. We now can edit single nucleotides, but as I told you, prime editing is coming on board rapidly, and while it is not as efficacious right now, we've tried it, and it really has less than 10% of the capacity of base editors, that in fact, as it advances, may have the opportunity to do more than just change a nucleotide, to insert many nucleotides or delete many nucleotides. There's a lot more to do in terms of vector development. We don't have the perfect vector. Delivering 2 adeno-associated viruses increases the risk of toxicity. We need to be able to have better designer vectors so that we can have large cargo inserted within them, akin to what we do with our mRNA COVID vaccines. Because if we can get those to be harnessed appropriately, I think we'll really be able to move this forward. There's lots and lots of opportunities, and I will end by telling you we are always looking for great, interested, enthusiastic trainees who want to come to Boston. We have a good time, and the lab would love to welcome all of you to Boston and to participate in these opportunities. So thank you for your attention. I really, again, thank the Zogbys for their incredible generosity of hosting me and the spirit with which this community they really so much enliven. Thank you.
Thank you so much for an amazing overview and major developments in this field. Thank you, really amazing. We'll open it for questions. Yes, please. Can you use a microphone?
Thank you so much for this incredible grand rounds. I really learned a lot. I just had a question because some of these mutations, don't just develop into HCM like the MY7. Some of them become non-compaction or dilated cardiomyopathy. So when I think about this, is if you're gene editing these genes, MY7 for instance, is there a possibility that this goes from HCM into somebody developing dilated cardiomyopathy or non-compaction cardiomyopathy just because of that gene editing?
Excellent question. So I think the audience out loud okay, would have heard that. We do not think that is a high risk for the following reasons. When we look at the mutations, especially the myosin mutations that can cause DCM and HCM, they are never at exactly the same location. When we see families that seem to have both phenotypes within a particular pedigree, we often find a second mutation. Hard to believe, but it's true. There are millions and millions of people in the world, and it does happen to mutations. The fact that the DCMs actually inhibit ATP hydrolysis, they don't accentuate it, I think provides really strong evidence that these are really acting at different locations with different effects. And so they would equally be appropriate for editing, but we would expect a very different phenotype. The other thing about editing to remember is the nucleotide that is targeted is going to be corrected back to the normal wild type sequence. It should not be changed to a different sequence. That would not be what we want. But you ask an important question that I didn't go into detail about, which is that when you target, the specificity for that nucleotide is not 100%. There are other nearby nucleotides in the guide that you direct the editors to that may, in fact, also be edited. So one will have to be very careful that those unintended edits, we call them bystander edits because they're in the same region, that those, in fact, are not going to lead to a different phenotype. And the good news is that we know a lot about myosin mutations and lots of mutations that we can predict. And so if that is what would be predicted to be an unexpected bystander edit, you need to design a better experiment. So not ready for clinical yet.
Thank you so much.
Yeah.
Just on that same note, in the same family, I follow families who have non-compaction.
Yes,
and they have
non-compaction.
And then they have HCM, and then they have other congenital disorders. So are you implying that there may be interaction of multiple genes that's causing?
Yes, I am implying that, and also hemodynamics. Non-compaction is a disorder, I look to my dear friend A.J. Marion over here, is a disease that is found with so many different genotypes that it makes me wonder if it is a consequence of something in the pathway that is being altered, not the direct effect of that mutation. Because even in a child who has non-compaction, you'll find that mutation that you think is myosin or something else, and they won't have non-compaction. There's a complex interaction that causes that, and I don't understand it, but I hope you could explain it to me.
Other questions?
AJ.
AJ?
Would like really to congratulate you. It must be exceedingly gratifying in the life of a physician scientist to make the first genetic discoveries, elucidate through elegant studies the molecular basis of the disease, and ultimately take those discoveries to develop a drug for a disease for which there was no treatment. Really, my sincere congratulations to you. It's a fantastic, beautiful journey. Seldom happens in the life of a physician scientist. Congratulations.
No. AJ,
and also
thank you.
it illustrates the impact of the fundamental discovery, whose significance at the time of discovery, many questions, and were known, and ultimately ends up to practice of medicine. I have one question for you about efficiency of base editing, especially with dual adenoviruses, which require the same virus, two viruses to get into the same myocyte to do. If each one is relatively, given that this is a heterozygous disease, half of the cells are mutant, half of the genes are mutant. So each virus affects about 75% of the myocyte. Two together to get into the same is about 50% chance. And that will at best lead to 25% correction of the underlying mutation. And given the efficiency, most of the time 3 to 5%, I wonder what is your experience on that?
Yeah, good questions. I would correct only one thing that you said, aside from the very kind compliment, which we know took a community to do, not me, not my lab, including you, your major contributions. But putting that aside, when I said 80% of the cells are corrected, that means that they got both viruses. Okay, so you can't correct it with half of an editor. Nothing happens. We know, we've proven that. Okay, so you have to have both within the same cell. The other reason why I'm very confident about that is called single cell sequencing. For many years, we would tissue sequence RNA and get a mixture that came from fibroblasts, endothelial cells, as well as cardiomyocytes. Now, when we do it's actually single nuclear sequencing, we know it's a cardiomyocyte based on all of the expressed genes. And those are where we see this impact. That's, again, due to the troponin T promoter, obviously. So we can get 80% of the cells to pick up both, okay? And we've done this over and over and over again. And so I'm absolutely confident in that. The problem, frankly, is the dosage that you do. So we use 10 to the 13th vectors per injection. So it's 2 times 10 to the 13th. And that's in a mouse, okay? Scale it up. And that's where we get into trouble. That's where the hepatotoxicity has appeared to begin to be seen. And in some of the early neurodegenerative disorders, we've seen lethality from high toxicity of the liver that appears to be an inflammatory response. The other thing, as many of you know, is there are many naturally occurring antibodies to AAV. AAV is not, in other words, going to be the final answer to this disease. Vectors are going to be the answer. And I really love, I mean, no one was in favor of COVID, but I was in favor of what COVID brought us, which is this, ability to deliver things in nanoparticles. I will tell you, our lab cannot get nanoparticles to permeate the heart cells yet, but we're trying. Got to modify them. So.
Thank you again for wonderful talk, and again, I'm too young in this field to acknowledge your contribution. One comment on the nano aspect before I go to the question, because we have seen certain nanoparticles actually permeate into the cardiomyocyte, and I think Guillermo Torre was the last author. I can share that paper. There were silica particles that we used before, but currently there are various nanoparticles that are being studied here that we've seen taken up inside the cardiomyocytes.
Perfect.
Maybe that's your right.
I have to tell you, we have a rule in my lab that if you go to a meeting, you have to come home with a new experiment. I just got mine.
Appreciate it. Well, my question comes from the clinical world, and then when you look at the genotypic inefficiency, and as you demonstrated, both in DCM and maybe from HCM, it appears there's an external influence that puts people from the environmental impact at a disadvantage. So my question is there a need or an opportunity for neurohormonal blockade in these individuals, especially when you see families with Titin mutation and young kids have it? Is there a need and opportunity to study that by putting beta blockers or ARBs or Entrestos?
The idea of penetrance, the concept of penetrance remains unsolved. And I would suggest that the reason we worked on myosins and C protein is the penetrance of those is very, very high. That's not true of Titin mutations, and we don't fully understand that. We have some ideas, but there's compelling evidence from a genetic point of view. Titin mutations are the most common genetic cause of dilated cardiomyopathy, end-stage, familial, et cetera. But we then, and our colleagues around the world have looked in other enigmatic dilated phenotypes, alcohol cardiomyopathy, chemotherapy-induced cardiomyopathy, peripartum cardiomyopathy. The prevalence of Titin mutation in those individuals is 10 to 20%. That's huge. And that says that they have a mutation and another event. And there's a lot of literature to support that the Titin mutations are very responsive to afterload reduction. So I like your idea. There's also evidence that when you look at a Titin carrier who is young and healthy and send them to the echo lab, he would say, normal, okay? And we send them to the MRI, they're normal. But if you then measure specifically dimensions where you're absolutely sure you're aligning all chambers across different individuals, some who have tightened and some who do not, they're all normal, but the walls are thinner and tightened, the volumes are increased, their ejection fraction is about 2.8 milliliters less than the healthy ones, the ones without the tightened mutations. They're all healthy. That suggests that we can only detect a range, okay? And it's not disease,
And you have
but
normal variability
right.
on top of that.
Exactly, exactly. So you're absolutely right. There's probably a subclinical phenotype there. I put that akin to before the hypertrophy, diastolic dysfunction is evident, hypercontractility is. There's remodeling going in those hearts. So if we had the capacity to intervene in those individuals, some of them we may spare what could happen. Some of them may not have anything happen. There was a recent, when we talked about polygenic risk scores very quickly, we know that our genomes and our hearts and our organs don't function due to one gene. So if you take tightened mutations and you overlay polygenic risk scores on top of it for DCM that remains unexplained, you can see a greater prediction of those that will have lower volumes. It was a Nature Communications, Huda's favorite journal, article that came out just a couple of years ago from one of our former trainees, Alex Bick. So it may be that we can tease out those people who we need to intervene on, but it's an important observation.
I have the microphone in front. Huda.
Beautiful talk. Really enjoyed it. Thank you so much. I'm going to go back to something really interesting, the small molecule, 461. You showed beautiful data in the mice when it was really early. It's prevented. That is pretty much, they did great. And that's an opportunity. The question I have, is it worth going into, I don't know the prevalence of this disorder, I don't know how many families there are, but take pre-symptomatic people, and even if you have to treat for a long time, that could become really a very valuable approach, and maybe when hopefully newborn screening comes online. So I'm just curious if there's such studies.
It's a wonderfully important opportunity, I agree. And in fact, we've all played that out with Marfan syndrome. We take children with Marfan syndrome and put them on beta blockers before they have aortic root dilatation because it's preventative. Just the diagnosis is worth taking the pill. The hiccup with 461 is, first of all, it was just FDA approved. It's not approved for kids or young people. And this is a real problem with big pharma. It's expensive. Big surprise, big surprise. So these are things that are not in my control, and I wish we could change, but that's the reality. Those trials, I'm sure, will go forward, but the biggest issue, I think, is the cost issue right now. And I understand about the cost of making drugs. I've learned a whole lot from our little company, Myocardia. But it is a big expense, and there has to be some way of recouping that. The question is how that should come, not from making patients pay that much.
Yes.
Not at all. So.
Go ahead, please.
That was a great talk, Christine, like always. I'm fascinated by the development of fibrosis in HCM, and my question is, does the problem with ATP hydrolysis, does it lead to a cascade of events that induces fibroblast activation, or is it an independent, indirect outcome of HCM process, cardiomyocyte abnormalities in cardiomyocyte relaxation?
Excellent, excellent question. The answer is A, we don't know the answer, and B, there are two kinds of fibrosis that we see in HCM, much more so than in DCM. We see interstitial fibrosis, where there is really not demise of the myocytes, but already you have this surrounding matrix that increases over time just because of that hypercontractility. We don't think it is related to the ATP, but it's actually the biophysical forces. Years ago, it was shown if you take cardiac fibroblasts and put them on a silastic membrane and just stretch them, that they will exude TGF-beta and they will increase collagen matrix deposition. So it could be the biophysical force that is actually causing that to occur. Whether that's true in vivo, we don't know.
The other kind of fibrosis, the focal fibrosis, we actually think is death of the myocytes. And we see that most commonly in the inner myocardium, and in particular in hypertrophied heart, and that probably is due in part to increased ATP requirements and the coronaries percolating in through a thickened myocardium get less blood than they need. And in explanted hearts, you can see sort of little foci in the endocardium of microinfarcts, which I think probably is a little bit of both. The fibrosis is a big deal. We are wondering if we could melt away the fibrosis, would that be good? In HCM, I think it would be. I'm not so sure about DCM.
Yeah.
But it might allow these other therapies to get in there too.
And collagen is a dynamic process, so fibrosis may not be, but
Yes.
if I could take the prerogative of asking the last question, excuse me. What was fascinating also in your talk is from a hemodynamics and cardiac conditions is, this is, I think, at least to me, first time where I see a condition that you demonstrated very nicely, that relaxation is impaired, yet contractility is increased. I haven't seen that in cardiology, because most of our diseases, you know, if relaxation is impaired, or at least if systole is impaired, diastole is.
Correct.
Is this specific to here, or have you seen it somewhere else?
No, I think it is specific to here, and it's also specific to the pathogenic mutations. When we take a heart tissue, we can't make a model of someone who doesn't have a mutation. So when we take heart tissues from mutation-negative HCM patients and measure SRX and DRX, we can't detect it. Those are, quite frankly, technically harder experiments to do because we have the little snippet of tissue from a myectomy. Maybe it wasn't so good, et cetera, et cetera. But I think it is very specific to these bizarre effects of increasing force production. And it also begs the question in my mind of these athletes who are physiologically training their hearts to always, always work hard and increase contractility. Over time, we see some of them fail. Is that because relaxation can't keep up? I don't know.
Because we've rationalized that this is reduced stress because of the hypertrophy of having a hyperdynamic ventricle. But you showed very nicely.
No, I think
that's a different phenomenon. Before we close, I'd like to acknowledge Dr. Jagat Narula. Dr. Narula was actually, here you go, great. Welcome to Houston, Dr. Narula. Dr. Narula was also was a Bill Winters lecturer 5 years ago. He also saw the light, similar to Dr. Ardehali, that coming to Houston from Mount Sinai is a good thing. So he will be at UT, and he will be the executive vice president for the system, in addition to the chief medical academic officer for University of Texas. So it's great to have you here. Again, thank you for an amazing presentation and a great discussion.